Welcome back to the Deep Dive, where we take a stack of your sources' articles, research your own notes, and unpack the most important nuggets of knowledge or insight.
Think of it as your personal shortcut to being well-informed, distilled from the material you've shared with us.
And we're here to guide you through that material, helping you navigate the details, identify the key insights, and understand what's truly crucial, especially as you delve into a new area or maybe reinforce some foundational knowledge.
Exactly.
Today, we are embarking on a deep dive into a fascinating and, I think, incredibly important topic, especially for you, currently immersed in studying dental sciences. We've got a stack of sources here focusing on the microscopic world living right there in our mouths, and its absolutely crucial, sometimes challenging role in both maintaining dental health and driving dental disease.
That's right. This deep dive is specifically tailored for you, exploring the fundamental biological players you'll be interacting with every single day in your future practice.
So what's our mission today based on these sources?
Well, our mission today, guided by these sources, is to explore this fascinating, sometimes challenging, microbial ecosystem of the oral cavity. We're focusing intently on how these tiny organisms contribute to the major dental infections you'll diagnose and treat, conditions like dental caries and periodontal disease.
Okay.
And we'll be specifically highlighting a critical, often overlooked group within this ecosystem, the non-sporing anaerobes.
Okay, non-sporing anaerobes. Got it. Let's unpack this and see what your sources reveal.
Let's do it.
So to start, let's really set the scene. What exactly is this environment we're talking about? Because the oral cavity isn't just an empty space, is it? It sounds like it's teeming with life.
Oh, it truly is. According to the sources, the oral cavity is one of the most heavily colonized parts of the entire human body. You can genuinely think of it as like a bustling, microscopic metropolis.
A metropolis, okay.
Yeah, it hosts an astonishingly diverse population of microorganisms. And we're talking about far more than just bacteria, although they are certainly major players. The sources note the presence of archaea, fungi, mycoplasmas, protozoa, and viruses, all coexisting and interacting within this single environment.
Wow. That's a lot of different types of life in one small area.
Mm-hmm.
The sources make a point of using specific terminology for this community, right, to distinguish things.
They do, and it's an important distinction for clarity. When we talk about the oral microbiome, the sources are referring to the totality of this community.
Okay, totality.
Yeah, that includes all the different types of organisms we just mentioned, bacteria, fungi, etc., plus their genetic information, and the entire oral environment itself, including all the physical and chemical conditions they live in and interact with. It's the complete ecological system.
Right.
In contrast, the term oral microbiota specifically refers to just the living microbes within that environment, so just the organisms themselves.
Okay, so microbiome is a whole ecosystem, organisms, genes, environment. Microbiota is just the collection of living organisms. Makes sense. And ideally, in a healthy state, this incredibly diverse community lives in balance.
Precisely. The sources emphasize that under normal circumstances, this vast community exists in a state of balance, a symbiotic equilibrium. The microbes live together, often benefiting each other, and even contributing positively to the host's health. It's a state of harmony within this microscopic city.
But here's where it gets really interesting, and I think directly relevant to the diseases you'll be focusing on. What happens when that harmony breaks down?
That's the critical shift. When this ecological balance is disrupted, the sources state that the system enters a state called dysbiosis.
Dysbiosis, okay.
And this imbalance is described as the fundamental root cause of many of the major oral diseases you will study, diagnose, and treat throughout your career. Conditions like dental caries, periodontal disease, and candidal infections. They're all rooted in this shift from health to dysbiosis.
So understanding this core concept, the difference between a healthy, balanced equilibrium and this state of dysbiosis is absolutely foundational for someone studying dental sciences.
Absolutely.
It kind of reframes how you think about oral health. It's not just about identifying a single bad bug and trying to eliminate it. It's more about understanding and, managing an entire complex, dynamic ecosystem.
That's exactly it. Your role in clinical practice isn't solely focused on eradication. It's heavily weighted towards restoring and maintaining that delicate balance. The sources make it really clear this ecological perspective is key.
Okay, so how do these teeming billions of microbes actually impact our oral health? What are the crucial roles they play according to the sources we're looking at?
Well, the sources outline several key aspects of their involvement. One of the most significant and likely already familiar to you from your studies is biofilm formation.
Which in the context of the mouth is essentially dental plaque.
Right. Exactly. The sources highlight that most bacteria in the mouth don't spend much time floating around freely as individual cells. That's the planktonic phase. They primarily exist in a biofilm phase, attached to the various oral surfaces.
Okay.
This structured, attached microbial community is what we commonly call dental plaque.
So plaque isn't just a random collection of bacteria. It's actually a highly structured, organized community.
Precisely. It's described as a complex, highly tenacious microbial community. And what makes it so sticky, so difficult to remove mechanically, is that the microbes are embedded within a matrix.
A matrix.
Yeah, and this isn't just any matrix. It's primarily composed of extracellular polysaccharides, or EPS. This sticky material, produced by the bacteria themselves, encases the microbes, binding them together and firmly anchoring them to the tooth surface.
And this matrix isn't just made of those polysaccharides and bacteria. There's other stuff in there, too.
No, you're right. The sources specify that this extracellular matrix also contains components from the host, like compounds from your own saliva, as well as living and dead bacterial cells and their metabolic products. It's a really complex biomaterial.
And this whole phenomenon of biofilms isn't unique to the mouth, right? It's a much broader biological principle.
It absolutely is. The sources emphasize that biofilms are ubiquitous in nature, and significantly, they point out that biofilms are associated with up to 65% of all human infections across the entire body, not limited just to the oral cavity.
Wow, 65%.
Yeah. It underscores their fundamental importance in infectious disease processes everywhere.
That's a staggering number. And it immediately highlights a major challenge we face in treating these infections, doesn't it?
It does.
The sources make it very clear that bacteria living within these organized biofilms, the sessile organisms, are generally significantly more resistant to antimicrobials compared to their free-living planktonic counterparts. Why is that? What makes them so tough in there?
Well, there are multiple mechanisms described in the sources contributing to this increased resistance. One is the physical barrier provided by that thick extracellular matrix we just discussed.
Okay, so it's like a shield.
Kind of, yeah. It can simply impede the penetration of antimicrobial agents, making it harder for them to reach the bacteria within. Another crucial factor is that bacteria undergo significant phenotypic changes when they live in a biofilm community.
Phenotypic changes, meaning they act differently.
Exactly. They alter their gene expression patterns, change their metabolic activity, and essentially adopt a state where they are less susceptible to the mechanisms by which many antibiotics work. It's almost like they activate a different survival mode inside that fortress.
Okay, so this is a critical takeaway for you listening as a dental science student. Plaque is not just some fuzzy surface contaminant. It's a highly organized, protected, and actively resistant microbial community. This inherent resistance of biofilm bacteria is a major clinical challenge you will encounter constantly, whether you're scaling a tooth, managing a carious lesion, or treating an infected root canal. You're dealing with a fortified enemy.
It really is, and it's why mechanical disruption of that biofilm, physically breaking it up and removing it, is so often a necessary cornerstone of treatment, even when antimicrobial agents are also being used.
Now, it's not all adversarial, is it? The sources also discuss some positive roles played by the microbes in our mouths. They're not all bad guys.
Absolutely not. Not all microbial activity is detrimental. The normal, resident oral flora provides a really important form of innate defense for the host, a concept known as colonization resistance.
Colonization resistance. What does that mean in a practical sense for the mouth? How does that work?
Well, it means that the existing healthy flora actively helps to prevent potential pathogens, whether they're new incoming microbes or opportunistic residents already there, from colonizing the mouth and establishing themselves in numbers sufficient to cause disease.
Okay, so they kind of guard the territory. And how do they achieve this resistance, according to the sources? What are their tactics?
They employ several mechanisms. They secrete metabolic byproducts, some of which have inhibitory effects on the growth of other microbes, essentially acting like, natural antibiotics. They also compete vigorously for essential resources. They simply consume the available nutrients that potential invaders would need to survive and multiply. Furthermore, they compete for attachment sites, those specific receptors or locations on oral tissues where microbes need to adhere. If the resident flora is occupying these sites, a pathogen can't really gain a foothold.
Makes sense.
And they can also actively modify the local microenvironment, for instance, by changing the pH or the oxygen levels, making it unfavorable for the growth of many pathogenic species.
That's fascinating. So the healthy flora isn't just passively present. It's actively protecting you from invaders.
It is, and for you as a dental science student, this means recognizing the healthy oral flora as your ally. Understanding this concept is key to appreciating why disturbing this delicate ecological balance, for example, by using broad-spectrum antibiotics that wipe out a lot of the beneficial normal flora, can sometimes inadvertently create an opening for opportunistic pathogens to take over and cause a new infection.
Right, which brings us back around to the idea of ecological balance and that shift to dysbiosis we discussed earlier.
Exactly. The sources reinforce this point. Oral diseases like caries and periodontal disease are outcomes of this disruption of the careful ecological balance within the oral microbiome.
And that disruption can be triggered by various factors, not just antibiotics.
Yes, the sources note it can be caused by intrinsic factors, which originate within the host, or extrinsic factors, which come from outside the host. The clear example provided, and a clinically relevant one, is definitely the use of broad-spectrum antibiotics.
Wow.
That's an extrinsic factor that can significantly deplete the numbers and diversity of the beneficial normal flora, creating an ecological niche, basically an opportunity for opportunistic pathogens to grow unchecked.
Like Candida albicans causing candidal infections, which you mentioned earlier. Thrush.
Precisely. Candida albicans is a classic example from the sources. It's a normal resident fungus, usually present in small numbers, typically kept in check by the bacterial flora through competition and inhibitory factors. But if you remove that bacterial competition with antibiotics, Candida can proliferate rapidly and cause infections like oral thrush or candidiasis.
This really emphasizes the interconnectedness of the whole oral ecosystem. As a future dental professional, your interventions, even seemingly localized ones like filling a cavity, can have downstream effects on these complex microbial populations. You're always influencing an entire living community.
Managing that ecosystem effectively is really a central challenge in clinical practice.
Okay, let's zoom in on one of the major diseases you'll undoubtedly be dealing with constantly.
Yeah.
Dental caries. The sources provide a pretty clear step-by-step journey of how this disease progresses, starting microscopically.
Yeah, dental caries is clearly defined in the sources as a chronic, non-communicable disease, and importantly, it's described as an endogenous infection.
Endogenous, meaning it comes from within.
Exactly. It originates from the dysbiosis of microbes already resident within the oral cavity, specifically those living in the plaque biofilm on the surfaces of the teeth, not from some outside invader.
And the fundamental process, boiled down, is bacteria eating sugar and producing acid that dissolves the tooth structure.
That's the core mechanism, yeah. It's the localized destruction of tooth tissues, enamel and dentine, resulting from the fermentation of dietary carbohydrates by bacteria within the plaque biofilm, which produces acids. The sources note it's a disease driven by the interplay of multiple factors. Host factors, like the tooth itself, its structure, the composition of saliva, diet, especially the frequency and amount of fermentable carbohydrates consumed, and crucially, the specific microorganisms within that plaque biofilm.
Okay, let's trace the typical progression of a carious lesion as described in your sources.
Yeah.
Where does it all begin? What's the first sign?
It begins with the initial lesion, often recognized clinically as a white spot lesion. This is the very earliest detectable stage of the carious process on the enamel. Clinically, you'd observe a well-demarcated area on the enamel that appears chalky white, often more visible after you dry the tooth surface.
And at this stage, the surface still feels smooth. No hole yet.
Yes, and this is a crucial point highlighted in the sources. At the white spot stage, the surface continuity of the enamel is still intact. What's happening microscopically is that the acids produced by the dysbiotic plaque microbiota are causing subsurface demineralization, basically, the loss of mineral ions from the enamel structure, specifically from the crystals beneath the surface layer.
This stage is really where prevention efforts can make the biggest impact, isn't it?
Absolutely. The sources strongly emphasize that at this white spot stage, the contradaceous process is reversible.
Reversible. Okay.
Yes. If the environment changes, if the pH is raised, acid production is reduced, and essential minerals like calcium and phosphate are available, say from saliva or fluoride treatments, remineralization can occur. The lost minerals can actually be redeposited, and the lesion can arrest or even appear to repair itself.
But if that doesn't happen, if the acidic attack continues?
Well, if the process continues and the subsurface demineralization progresses and eventually undermines the surface enamel, that weakened surface layer can collapse. This leads to cavitation.
The hole.
Right. A physical hole, a cavity, forms in the enamel. Once cavitation occurs, The sources are clear. This indicates irreversible disease, at least in terms of structure. You cannot simply remineralize enamel back once it's physically lost like that.
And from the enamel cavity, it just spreads deeper into the tooth?
Yes. If left untreated, the cavitation provides a direct pathway for bacteria and acids to progress deeper into the tooth structure, into the dentine. Dentine is the layer beneath the enamel. The sources note that caries progression in dentine is often faster than in enamel because dentine is less mineralized and contains microscopic tubules that bacteria can penetrate more easily.
Okay.
This spread through the dentine can eventually reach the central part of the tooth containing the nerves and blood vessels, the dental pulp.
Which leads to infection and pain, right, within the pulp.
Right. The dental pulp can become infected via the advancing calcareous lesion. This leads to inflammation of the pulp tissue, which we call pulpitis. If this pulpitis is not treated promptly, it can become irreversible and ultimately lead to pulp necrosis, the death of the pulp tissue.
Necrosis? Dead tissue inside the tooth?
Yes. And at this advanced stage within the tooth, the sources describe a complex microbial biofilm forming on the inner surfaces of the root canals that were once filled with vital pulp tissue.
And the sources detail a pretty serious potential consequence when infection spreads beyond the tooth itself.
They do. When the pulp is fully necrotic and heavily infected with a biofilm, the bacteria and their toxic products can extend out from the tip of the tooth root through the little opening called the apical foramen and into the surrounding bone and tissue, the periapical tissue. This results in a dentoalveolar abscess.
Okay, so a collection of pus at the end of the root. What's the microbial picture like in these abscesses? Is it just one type of bacteria?
No, the sources consistently describe these dentoalveolar abscesses as typically polymicrobial endogenous infections, meaning they involve multiple species of bacteria already resident in the oral cavity.
Right.
And a very significant finding emphasized is that these infections predominantly involve strict anaerobes.
Ah, the anaerobes again.
Exactly. This makes perfect biological sense, as the necrotic pulp tissue and the center of an abscess cavity are very low oxygen environments where these bacteria thrive.
And the danger of these infections isn't limited just to losing the tooth, right? They can spread.
They absolutely can. The sources highlight that these dental alveolar infections have the potential to spread to surrounding tissues and anatomical spaces. This includes spreading into the deeper medullary spaces of the alveolar bone, potentially leading to osteomyelitis bone infection.
Yeah.
They can also spread to nearby areas like the maxillary sinus, and although thankfully rare, they can sometimes track along fascial planes into deeper spaces of the head and neck, or even in severe cases disseminate via the bloodstream or adjacent structures to the central nervous system, leading to really serious complications like brain abscesses or meningitis.
Wow. That's a stark reminder of the potential severity. So for you as a dental science student listening, this stage-by-stage breakdown from the sources is your essential roadmap. It's how you diagnose and understand the progression and severity of caries. Recognizing the reversibility of that initial white spot lesion is a major clinical opportunity for preventative intervention and patient education. And understanding the potential for widespread, even life-threatening infection from an advanced lesion underscores the critical importance of treating caries promptly and effectively.
It really shows the transition, doesn't it? From what starts as a localized mineral balance issue to potentially a significant systemic health risk if left unchecked.
Definitely. Okay, so we know dental caries is driven by bacterial activity within plaque, but which specific bacteria are considered the main culprits? The sources discuss evolving theories over time, but ultimately point to key players with specific properties that make them, well, bad actors in this story.
That's right. While the microbial community involving caries is definitely complex and involves many species, the sources highlight certain species as having disproportionate significance in initiating and progressing the disease.
And the ones most strongly linked to actually initiating the lesions, starting the trouble.
The leading contenders, widely considered major agents in the initiation of dental caries, are the mutans Streptococci, specifically Streptococcus mutans and Streptococcus sobrinus.
Mutans Streptococci.
Okay.
What is it about these particular species that makes them so effective at causing cavities? What's their toolkit?
Well, the sources detail their powerful set of cariogenic properties. First, their ability to produce extracellular polysaccharides, EPS, especially when there are dietary carbohydrates around, particularly sucrose. They produce large amounts of this sticky material.
The matrix stuff again.
Exactly. And it's crucial because it allows them to adhere very tenaciously to the smooth enamel surface and also stick to other bacteria, basically forming and reinforcing the core structure of the protective plaque biofilm.
So they're like the master builders of the plaque. They build the scaffolding.
You could say that, yeah. Second, they are highly acidogenic. They are very efficient at metabolizing fermentable carbohydrates like sugars and producing significant quantities of organic acids, primarily lactic acid, as a metabolic byproduct. This process rapidly lowers the pH within the plaque biofilm right at the tooth surface.
Okay, they make a lot of acid quickly.
Yeah.
And most bacteria don't tolerate very low pH environments, but these guys do.
That's their third critical property, acidoricity. They are not only acidogenic, acid-producing, but also remarkably tolerant of acidic conditions. They can survive and continue to be metabolically active, including producing more acid even at very low pH values, like below 5.5, where many other oral bacteria would be inhibited or even killed off.
Wow.
This ability allows them to sustain the acidic attack on the enamel surface that's necessary for demineralization.
And they have a way to keep producing acid even between meals when you're not eating sugar.
They do. They also have the ability to produce intracellular polysaccharides, IPSs. They can take up dietary sugars when available and store them internally as glycogen-like polymers. These internal food reserves allow them to continue fermenting sugars and producing acid even when dietary carbohydrates aren't immediately available from the environment, effectively prolonging the period of low pH attack on the tooth.
Man, they sound like they're perfectly evolved to attack the tooth surface. Like little acid factories with backup generators.
They are exceptionally well adapted to this niche, especially in the presence of frequent sugar exposure, and the sources note a strong clinical correlation. Higher numbers of S mutants in saliva and plaque are often associated with increased prevalence and incidence of dental caries.
Okay, so S mutants and Sobranius are major initiators. Who else plays a significant role in the caries process, according to the sources?
Lactobacilli are also very important players mentioned frequently. They are often isolated, particularly from deep carious lesions and right at the advancing front of dental caries. Like the mutant Streptococcus, they are both acidogenic and aciduric.
So they also produce acid and tolerate low pH. How is their role different from S mutants?
Yes, they do both. While historically they were sometimes thought to be the primary cause of all caries, their role is now understood, according to the sources, to be more significant in the progression of established enamel lesions and especially the advancement of dentinal caries, rather than necessarily initiating the earliest lesions on smooth enamel.
So they're more involved later in the process.
Kind of. Because they thrive in acidic environments, once the pH has been lowered by initial colonizers like S mutants, lactobacilli can really proliferate and contribute significantly to the ongoing demineralization process, especially deeper in the lesion. The sources also mention that salivary lactobacilli counts can sometimes be used as an indicator of a patient's high dietary carbohydrate intake and their overall caries activity level.
Interesting. Are there specific bacteria linked to root surface caries? Because that's a bit different from enamel caries, right? It happens on the root when gums recede.
Yes, it is different, and the sources mention specific bacteria. Actinomyces species, particularly Actinomyces naslundii and Actinomyces odontolyticus, are implicated in root surface caries. Unlike enamel, root surfaces, which are covered by cementum, are softer and start to demineralize at a higher pH. These Actinomyces species are gram-positive rods that are major components of dental plaque generally, and their numbers tend to increase during gingivitis, which often precedes the root exposure needed for root caries to occur. Rothia dentacariosa is another species mentioned in relation to root surface caries in the sources.
Okay, now this picture seems to be mostly about microbes producing acid. Are there any microbes in plaque that actually counteract the acidic effects? Maybe help things out?
Yes, there's an interesting group that is considered potentially beneficial in the specific context. Veillonella species, these are gram-negative anaerobic cocci. The sources describe them as potentially benevolent or even protective in the context of caries progression.
Benevolent. How can a bacterium in plaque be good for your teeth in this situation?
Well, because they utilize lactic acid. Lactic acid is the primary, most damaging acid produced by the main cariogenic bacteria like S. mutans and lactobacilli. Veillonella species, however, actually metabolize this lactic acid and convert it into weaker organic acids like propionic and acetic acid, which are much less effective at demineralizing tooth structure.
Ah, so they eat the bad acid.
Essentially, yes. By consuming lactic acid, they effectively help to remove it from the plaque environment, potentially raising the pH slightly and counteracting the demineralization process to some degree.
That's a great example of the complexity of the ecosystem. It's not just good guys and bad guys. Some microbes are actively working against others. And the sources note that with modern techniques like DNA sequencing, we're identifying even more players involved than we used to know about.
Absolutely. Traditional culture methods could only identify a fraction of the microbes present. Modern molecular techniques, especially next-generation sequencing, NGS, have revealed a much more diverse microbial community associated with dental caries, including many species that are difficult or impossible to culture in the laboratory.
Like who? Any examples given?
Yeah, examples given in the sources include species from the genera Edipobium, Dialister eubacterium, Olsonella, and Scardovia. Their specific roles in the caries process are still being actively researched and defined, but we now know they're often present in carious lesions.
Okay, so for you as a dental science student, this section provides the key microbial players and caries and, crucially, why they are important, their specific properties and mechanisms.
Right.
Understanding why S. mutans is such a potent cariogen, its EPS production, acidogenicity, aciduricity, IPS storage, and how lactobacilli contribute significantly to lesion progression, particularly in dentine, is vital. It helps with targeted prevention strategies, risk assessment, and treatment planning. And recognizing the nuanced role of organisms like Veillonella adds important context to that whole ecological perspective we talked about.
It's definitely about the interplay of specific functions within that dynamic community, not just the presence or absence of one species.
Right. Okay, let's dive deeper now into a particularly important group highlighted in the sources, one that's crucial for understanding many severe oral infections. The non-sporing anaerobes. You mentioned them earlier.
Yes, this group is absolutely central to many of the deeper, more advanced oral infections you will encounter in clinical practice. As their name indicates, these are bacteria that do not form environmentally resistant endospores like some other bacteria do.
Okay, no spores. And where do we find them thriving, generally? Where do they like to live?
Well, they are widespread in nature, but they are particularly abundant in the human body, especially in locations where oxygen levels are low or completely absent.
So they require specific oxygen conditions to grow. They don't like oxygen.
Exactly. The core group are obligate anaerobes, meaning they require very low or zero oxygen to grow and reproduce. Oxygen is often toxic to them.
Okay.
The sources also mention some that are microaerophilic, capable of growing in low oxygen concentrations, and others that are aerotolerant, meaning they can tolerate exposure to oxygen for a period, but still prefer or require anaerobic conditions for sustained growth.
And how prevalent are they compared to bacteria that like oxygen, the aerobes, or facultative anaerobes?
The sources note their significant prevalence in specific body sites. In environments like the large intestine, for instance, obligate anaerobes outnumber facultative anaerobes by as much as 1,000 to 1.
Wow, 1,000 to 1.
Yeah. And on skin and mucous membranes, including the oral cavity, they typically outnumber facultative anaerobes by a factor of maybe 5 to 10 times. The oral cavity, with its diverse microenvironments like the deep gingival crevice, periodontal pockets, and the interior of a necrotic tooth, offers many perfect low-oxygen niches for these organisms to flourish.
And the sources mention a shift in how the scientific and clinical communities view these microbes over time. They weren't always seen as troublemakers.
That's a crucial historical perspective. Many of these non-sporing anaerobes were once considered relatively harmless commensals, just part of the normal flora that didn't cause problems. However, as research advanced, particularly with improved culture techniques for anaerobes and the advent of molecular methods, they are now clearly recognized as opportunistic pathogens.
Opportunistic pathogens, meaning they cause disease only when the conditions are right.
Precisely. While they live normally in many sites without causing harm, under certain conditions like when host defenses are compromised, when tissue is damaged providing nutrients and anaerobic conditions, or when the local environment becomes favorable, like becoming anaerobic due to inflammation or necrosis, they can proliferate, invade tissues, and cause serious infections.
So for you as a dental science student, understanding this group is non-negotiable. Non-sporing anaerobes are critical, often dominant players in many of the deep advanced infections you will see, such as dental-alveolar abscesses, severe forms of periodontitis, and infected root canals.
Absolutely.
Knowing their oxygen requirements is absolutely fundamental to understanding where in the mouth or tooth these infections typically originate and how they progress into those low-oxygen anaerobic environments.
Their specific environmental needs really dictate their pathogenic potential in different oral sites.
Okay. The sources provide a pretty detailed breakdown of some of the most clinically relevant non-sporing anaerobes you'll encounter in dentistry. They classify them by their Gram-stain reaction and morphology, which are classic ways bacteria are grouped. Let's walk through some of these key players and what the sources say about them.
Right. It's a bit of a list, but these are important names to start recognizing, starting with the gram-positive Kochi. So, round bacteria that stain, purple with gram stain. A key genus here, particularly associated with infections like abscesses, is Peptostreptococcus, sp.
Peptostreptococcus.
Yes. These are obligate anaerobes and, as we discussed earlier when talking about dentoalveolar abscesses, they are frequently isolated from these predominantly anaerobic purulent infections.
And the sources note a taxonomic change regarding Peptococci, something about names changing.
Yes, it's a point of clarification they make. Historically, many anaerobic gram-positive cocci were grouped under the genus Peptococcuses. However, based on genetic analysis, most of these have now been reclassified into the Peptostreptococcus genus. Peptococcus niger is mentioned as one of the few exceptions that remains in the original Peptococcus genus. Just something to be aware of if you see older literature.
Okay, good to know. Any other gram-positive cocci highlighted?
Stomatococcus mucilaginous is mentioned, though the source notes it's actually a facultative anaerobe, not strictly anaerobic like the others in this section, but it's found in the mouth, mainly on the tongue and in the gingival crevice. It's not generally considered a major opportunistic pathogen. Also, Granulicatella species are discussed.
Granulicatella?
Yeah, these were previously known as nutritionally variant streptococci because they have picky growth requirements. They are gram-positive cocci, facultatively anaerobic, and need specific nutrients like pyridoxal or L-cysteine to grow in culture. The sources note they are non-motile, catalase negative, and oxidase negative. These are biochemical characteristics useful for identification in the lab, indicating specific metabolic pathways related to oxygen detoxification and energy production.
Okay. Moving on to the gram-positive bacilli, or rods. Rod-shaped bacteria that stain purple.
A very important genus here, which we also mentioned earlier in the context of caries, is Actinomyces species. These are true bacteria, although their filamentous branching growth pattern once led researchers to mistakenly classify them as fungi. They look a bit like fungi under the microscope sometimes.
Right.
They are typically microaerophilic or anaerobic. The sources emphasize they are a major component of dental plaque, and their numbers increase significantly in conditions like gingivitis.
And specific species are linked to specific infections.
Right.
Yes. Actinomyces israeli is a key human pathogen causing actinomycosis. These are chronic, slow-developing granulomatous infections, often affecting the cervicofacial region, the head and neck. These infections are characterized by the formation of macroscopic yellowish aggregates of bacteria called sulfur granules, which you might see in pus.
Sulfur granules, okay.
In the mouth itself, Actinomyces odontolyticus is linked to early enamel demineralization and caries progression, and Actinomyces nasalindii is implicated in root surface caries and gingivitis.
We discussed lactobacilli with caries. They also fit here as gram-positive rods, correct?
Yes, lactobacilli are gram-positive, mostly rod-shaped bacteria. As we discussed, they are typically facultative anaerobes, but they are strongly acidogenic and aciduric, playing a significant role in the progression of dentinal and root surface caries. While often facultative, they are a notable group of gram-positive rods found in these anaerobic environments within deep lesions.
How about Propionibacterium? Heard that name before, I think related to skin.
Yes. Propionibacterium species are gram-positive bacilli and strict anaerobes. They are often slow-growing. Propionibacterium acnes, which the sources note now includes the former species P. propionicus, is indeed found on skin but also on teeth, particularly in caries dentine. It's also isolated from periodontal and dentoalveolar abscesses, typically as part of a mixed infection involving multiple species.
Okay. In Eubacterium, you mentioned them in relation to biofilms earlier.
Yes, Eubacterium species are described as pleomorphic, meaning they vary in shape, and can be gram-variable rods or filaments. They are obligatory anaerobes. The sources state they are found in plaque biofilms and calculus, Tartar. They are implicated in both caries and periodontal disease and can constitute a significant proportion, sometimes over 50% of the anaerobes found in deep periodontal pockets. Eubacterium uri is specifically highlighted for its role in forming those distinctive corncob structures within dental plaque biofilms, where cocci like streptococci attach all along the length of the filamentous eubacterium rods.
Corncob structure is interesting visual. Any other gram-positive rods of note from the sources?
Bifidobacterium dentium is mentioned as a gram-positive strict anaerobe commonly isolated from plaque biofilms, although its specific pathogenic role isn't clearly delineated in the provided material. And Rothia dinocariosa, which we linked to root surface caries earlier, is also a gram-positive branching filament. It's worth noting, as the sources do, that while discussed in the context of oral flora in plaque, Rothia dentacariosa is often described technically as a strict aerobe, needing oxygen, which makes it distinct from many other bacteria in this list that prefer low oxygen. It's also occasionally associated with systemic infections, like infective endocarditis if it gets into the bloodstream.
Okay, quite a variety there. Let's move to the gram-negative cocci. Round bacteria that stain pink or red. We've already met one, the benevolent one, right?
Yes, exactly. Veillonella species. These are obligate anaerobic gram-negative cocci. They are very commonly found in oral samples, especially dental plaque. As we discussed, the sources deem them potentially benevolent or protective in the context of caries due to their ability to utilize lactic acid produced by cariogenic bacteria, converting it into weaker acids and potentially helping to moderate plaque pH. They have no known pathogenic potential in the oral cavity according to the source material.
Good to have some potentially helpful ones in the mix. And Neisseria were mentioned as early colonizers.
Yes, Neisseria species, specifically the commensal ones like Neisseria subflava, N. mucosa, and N. sicca, are common normal flora in the oral cavity, nose, and pharynx. The sources point out that they are among the earliest bacterial colonizers of a clean tooth surface attaching to the salivary pellicle that forms first. An interesting physiological point is that they're aerobic, they use oxygen, and can actually consume residual oxygen present on the tooth surface. By doing this, they help to lower the redox potential and create more favorable anaerobic conditions deeper within the developing biofilm for the strict anaerobes that arrive later.
So they're kind of preparing the environment for the strict anaerobes. Clever. Generally considered non-pathogenic in the mouth, though.
Generally, yes. The oral commensal Neisseria are considered non-pathogenic in the oral cavity.
Okay, now, the gram-negative bacilli group, the pinkard staining rods. This category contains some of the most infamous periodontopathogens, doesn't it? The ones really strongly linked to gum disease.
It certainly does. This category includes some absolutely critical players in serious oral infections. First up, the Porphyromonas species. These are obligately anaerobic, gram-negative rods, often pleomorphic in shape, meaning variable shape.
Yeah.
Porphyromonas gingivalis stands out. It's highlighted as a key pathogen, particularly associated with chronic periodontitis and found frequently in dental alveolar abscesses.
P. gingivalis. What makes it so virulent, according to the sources? What's its arsenal?
Well, the sources describe it as highly virulent due to its production of a formidable array of damaging factors. This includes potent proteases, enzymes that break down host proteins like collagen and immunoglobulins. It also produces hemolysin, which laces red blood cells, collagen-degrading enzymes contributing to connective tissue breakdown, and various cytotoxic metabolites that can directly harm host cells.
Wow.
Its capsule and its fimbria, hair-like appendages, are also highlighted as important virulence factors that aid in its attachment to host tissues and its ability to evade the host's immune defenses. P. endodontalis is another species in this genus, commonly isolated from infected root canals.
Okay, P. gingivalis sounds like a major player. Next, Prevotella species, related.
Also obligately anaerobic, gram-negative pleomorphic rods, and often found alongside Porphyromonas. The human oral cavity is noted in the sources as being a primary ecological niche for many Prevotella species. Prevotella intermedia is strongly associated with periodontal disease and shares some virulence properties with P. gingivalis. It's found in periodontal pockets and dental plaque biofilms and is implicated in chronic periodontitis, dental alveolar abscesses, and also pericornitis, inflammation around wisdom teeth.
And Fusobacterium, they have a distinctive shape, right?
Yes. Fusobacterium species are non-sporing, anaerobic, spindle-shaped rods that are normal inhabitants of the oral cavity. Fusobacterium nucleatum is the most common species isolated from the mouth. It's considered a critical bridging organism in biofilm development because it can stick to both early and late colonizers, helping different species co-aggregate and build up the plaque structure.
Ah, like a connector.
Exactly. It's also a key component of the fusosporicatal complex, a specific microbial grouping strongly linked to acute necrotizing ulcerative gingivitis, ANUG, also known as trench mouth or Vincent's angina, as well as being found in advanced periodontal disease. Fusobacteria can also produce volatile sulfur compounds like hydrogen sulfide and ammonia, which contribute significantly to halitosis or bad breath.
Okay, ANUD and bad breath, got it. Are there other significant gram-negative rods associated with periodontitis mentioned?
Yes, several others. Tannerella forsythia, which used to be known as Bacteroides forsythus, is another obligate anaerobe recognized as a significant periodontal pathogen. It's noted for being difficult to culture in the lab, often requiring a long incubation time. Interestingly, its growth is actually enhanced when it's co-cultivated with Fusobacterium nucleatum.
So they help each other out.
Seems like it. Its endotoxin, part of the gram-negative cell wall, and other metabolic products contribute to its virulence.
And wasn't there one species specifically linked to aggressive forms of periodontitis, especially in younger people?
Correct. That's Aggregatibacter actinomycetum comitans, often abbreviated as AA. This organism is a microaerophilic or capnophilic, meaning it grows best in a CO2-rich atmosphere. Gram-negative coccobacillus, sort of a short rod shape. The sources identified as a key pathogen, specifically in localized aggressive periodontitis.
And what makes A so aggressive?
It produces several potent virulence factors, notably leukotoxin, which is toxic to host white blood cells, particularly neutrophils, damaging the immune defense. It also produces proteases that cleave immunoglobulin G, IgG, further impairing host immunity. It's found in deep periodontal pockets and is also associated with peri-implant diseases around dental implants.
Leukotoxin. Sounds nasty. Any others in this gram-negative rod category mentioned in the sources?
A couple more. Capnocytophaga species are also discussed. These are CO2-dependent gram-negative fusiform, spindle-shaped rods that exhibit a characteristic gliding motility across surfaces. They are isolated from periodontal pockets, plaque, and mucosal surfaces. While some species are considered part of the normal flora, the sources note they can cause opportunistic infections, particularly in immunocompromised patients, and are associated with a destructive periodontal disease.
Gliding motility, interesting.
And finally, Eikenella species like Eikenella corrodens. These are gram-negative coccobacilli that are commensals of the oral cavity. While often normal residents, the sources highlight their notable involvement in infections resulting from human bite injuries. They are often found in those wounds. They can also cause serious systemic infections like endocarditis, meningitis, pneumonia, and wound infections if they enter the bloodstream, particularly in susceptible individuals.
Okay, that covers the gram-negative rods. Distinct from these, based on shape and staining, the sources also discuss spirochetes. What are they like?
Spirochetes are a very distinct morphological group. They are helical, spiral-shaped organisms and highly motile, often moving with a corkscrew motion. They stain poorly with standard gram stains, so special microscopy techniques like dark ground microscopy, which makes them appear bright against a dark background, or immunofluorescence are typically needed to visualize them effectively. They are strictly anaerobic or microaerophilic.
And which ones are relevant to oral infections?
The key genera include Treponema species. Oral treponemes, such as Treponema denticola and T. vincentia, are primarily found in the gingival margin and crevice, especially in diseased sites. T. denticola is particularly highlighted for its highly proteolytic activity, meaning it's very effective at breaking down proteins, contributing to tissue damage, and its strong association with ANU and advanced periodontal disease as part of that specific fusospirochaetal complex we mentioned earlier, along with Fusobacterium nucleatum.
Ah, the fusospirochaetal complex again.
Exactly. The sources note they are generally sensitive to common antibiotics like penicillin and metronidazole, which is clinically useful. Borrelia species are also mentioned, with Borrelia vincenti being an obligate anaerobe and normal commensal that is also associated with Vincent's angina, ANUG. Leptospira interrogans, which causes leptospirosis, a systemic illness often from animal urine, is listed as a spirochete genus, but is less directly involved in the mechanisms of routine oral infections discussed in these sources.
Okay. Are there any other non-bacterial anaerobic residents of the oral cavity that the sources mention? Things that aren't bacteria but still live there without oxygen?
Yes, although they typically constitute a much smaller component compared to the vast numbers of bacteria. Archaea are mentioned, specifically methanogenic archaea like Methanobrevibacter oralis. While their specific role isn't fully clear, the sources note that their numbers can sometimes vary, potentially increasing, in individuals with periodontitis.
Okay. Anything else?
Also, oral protozoa, including Entamoeba gingivalis and Trichomonas tenax, are mentioned. These are single-celled eukaryotic organisms, and they are strict anaerobes found in the oral cavity, often associated with poor oral hygiene. However, the sources state they are currently considered harmless saprophytes, meaning they live off dead or decaying matter, and their direct role in causing periodontal disease remains unproven based on the evidence presented.
Wow, okay. That's a truly comprehensive look at the microbial landscape, especially the anaerobes. For you as a dental science student listening, this detailed catalog from the sources is invaluable. It directly links specific microbes, their characteristics like oxygen needs, gram stain, shape, and especially their key virulence factors like P. gingivalis proteases or A. actinomycetum comitans leukotoxin to the specific diseases and clinical presentations you will encounter. Knowing which species are notorious for conditions like chronic periodontitis, P. gingivalis, aggressive periodontitis, A. actinomycetum comitans, or A and U, that Fusobacterium nucleatum and treponema complex, is absolutely essential. It helps guide your diagnostic process and inform your treatment decisions.
Understanding these specific players and their capabilities is really fundamental to understanding the infections themselves.
Okay, let's step back slightly and look at the bigger picture again, how all these microbes function within the oral environment, especially within those crucial biofilms we keep mentioning.
Right. As the sources emphasize, the oral cavity is this complex ecosystem, and the non-sporing anaerobes we've just detailed thrive in specific low-oxygen niches within it. The most prominent example is definitely the non-shedding surfaces of the teeth, where they form those dynamic structured biofilms plaque, but also the low oxygen environment of the gingival crevice and, especially in disease, the periodontal pockets.
And dental plaque is the perfect natural example of this biofilm structure, isn't it?
It is. Dental plaque is the classic model of a natural biofilm in the human body, and the sources clearly describe it as the primary agent for initiating and progressing both dental caries and periodontal disease. And it's good to remember, as the sources point out, the oral microbiome encompasses far more than just bacteria. It includes fungi, the mycobiome, and viruses, the virome too, all interacting within this environment, although our focus here has been mainly bacteria.
We touched on this earlier, but it seems worth reinforcing. Identifying all these microbes has been a challenge historically because so many of them are hard to grow in the lab, right?
That's a very significant point highlighted in the sources. A large proportion of these oral microbes, particularly many of the strict anaerobes we just discussed, are uncultivable using predictional laboratory methods.
Uncultivable.
Well, they're often very fastidious, meaning they have complex nutritional requirements or extremely specific atmospheric requirements that are hard to replicate perfectly on a culture plate. This difficulty in culturing is precisely why the advent of molecular techniques, especially sequencing the 16S rRNA gene, which is like a barcode for bacteria, has been so revolutionary for microbiology.
How so?
These methods allow us to identify bacterial species based on their unique genetic sequences directly from a sample, like plaque or saliva, revealing the true diversity of the oral microbiota, including hundreds of species we couldn't previously detect because we couldn't grow them.
So we know much more about who's actually living there now. And the formation of these biofilms isn't just a random clumping of bacteria. It's a specific step-by-step process, according to the sources.
It is indeed a dynamic, structured process. The sources describe it beginning with the absorption of host proteins and glycoproteins from saliva onto the clean tooth surface, forming what's called the acquired salivary pellicle. It's like a conditioning film.
Okay, a base layer.
Exactly. Then, early bacterial colonizers, often gram-positive cocci and rods like certain streptococci, S. sanguinis, S. oralis, and actinomyces species, attach specifically to this pellicle. These are the pioneers.
And these early colonizers then change the environment, allowing others to join the party.
Precisely. The pioneer species modify the local environment. For example, as we mentioned with Neisseria, they might consume oxygen, creating conditions that favor the attachment and growth of later colonizers, which include gram-negative cocci and rods. The process continues with secondary colonizers like filamentous organisms, fusobacteria, remember the bridging organism, and spirochetes, which attach to the already present bacteria, often through very specific molecular coaggregation mechanisms, building up the complex, mature polymicrobial biofilm structure layer by layer.
And we even see specific structural arrangements within the mature biofilm, like those corn cobs.
Yes, like the corncob structures mentioned with bacteria, such as Eubacterium uri, where gram-positive cocci bind all along the surface of the filamentous bacteria, creating a distinctive three-dimensional architecture within the plaque. There are other arrangements too, like test tube brushes. It's quite organized.
Fascinating. And the major clinical takeaway here circles back to something we stressed earlier, the incredible resistance of these biofilms.
Absolutely. It's worth reiterating because it's so fundamental to treatment success or failure. Microorganisms living within these structured biofilms are generally significantly more resistant to both host immune defenses, like white blood cells trying to engulf them, and applied antimicrobials, like antibiotics or mouth rinses, compared to their free-living planktonic counterparts.
And why is that, again, briefly?
It's a multifaceted phenomenon. It includes the physical barrier of the protective extracellular matrix, slower growth rates of bacteria deep within the biofilm, altered gene expression leading to resistance mechanisms being turned on, and the presence of persister cells, which are metabolically dormant and tolerant to antibiotics.
Okay, so for you as a dental science student, understanding the biofilm life cycle, its complex structure, and its inherent resistance is absolutely key. It directly explains why mechanical debridement, physically removing the bulk of that biofilm mass using instruments like scalers or curettes, is often the primary and most essential step in treating plaque-related diseases like caries and periodontitis. Even when you might also be using antibiotics, you usually have to dismantle that protective fortress first to have a real impact.
The physical removal is often critical to overcoming that inherent biofilm resistance. You can't just rely on chemicals alone in most cases.
Right. Let's make this even more concrete now by explicitly connecting the specific microbes we've discussed to the various dental infections you will be diagnosing and treating as laid out in your sources. Let's put the bugs with the diseases.
Agreed. Starting again with dental caries. As a recap, the sources point to mutans streptococci, S. mutans, S. sabrinus, as key initiators, particularly on smooth enamel surfaces, driving that early demineralization due to their efficient acid production and biofilm formation capabilities. Lactobacilli are strongly associated with the progression of established lesions, especially deeper into dentine and on root surfaces, precisely because they thrive in the increasingly acidic environment created by the initial acid producers.
And actinomyces.
Actinomyces species, like A. naselindii, are also specifically linked to the initiation and progression of root surface caries. The prevailing ecological plaque hypothesis, which the sources mention, suggests that it's the frequent exposure to fermentable carbohydrates that drives down the TH, selecting for and allowing these acidogenic and aciduric species to dominate within the plaque, that's shifting the ecological balance towards demineralization and disease.
Okay, that's caries. What about periodontal diseases, diseases affecting the gums and supporting bone?
Here, subgingival plaque biofilms, the plaque accumulating below the gum line, are considered the primary etiology. The sources describe a significant microbial shift as relatively healthy gingiva or mild gingivitis progresses to destructive periodontitis. The community changes dramatically.
How does it change?
It shifts from being dominated by gram-positive and facultative anaerobic species, like certain streptococci and actinomyces, associated with health or simple gingivitis, to a dramatic increase in the proportion and absolute numbers of obligately anaerobic gram-negative bacteria in the deeper periodontal pockets that form.
And this shift involves those specific, more pathogenic groups we listed earlier, like the Porphyromonas and Prevotella.
Yes, exactly. This includes the so-called red complex and orange complex organisms, often including the black pigmented anaerobes like Porphyromonas gingivalis and Prevotella intermedia due to their colony appearance on blood agar, as well as Spirochaetes like Treponema denticola and Tannerella forsythia. The key gram-negative periodontopathogens consistently highlighted in the sources are P. gingivalis, P. intermedia, Tannerella forsythia, Aggregatibacter, Actinomycetum comitans, Fusobacterium nucleatum, and sometimes Capnocytophaga species.
And specific periodontal conditions are linked to certain microbial combinations or individual species, right? It's not always the same mix.
That's right. The sources detail several specific associations. Acute necrotizing ulcerative gingivitis, ANUG, that really painful, rapidly destructive condition, is described as a specific anaerobic polymicrobial infection. It's primarily caused by the synergistic action of fusobacteria, especially F. nucleatum, and oral Spirochaetes, Treponema species like T. denticola and T. vincentia, forming that characteristic fusospirachaetal complex visible under microscopy.
Okay, ANG has a specific microbial signature. What about pericoronitis, the inflammation around wisdom teeth?
Pericoronitis is also frequently caused by a mixture of normal oral bacteria that get trapped under the flap of gum tissue, the operculum, over a partially erupted tooth. This mix often includes strict anaerobes like Prevotella intermedia, Fusobacterium species, and anaerobic streptococci, which thrive in that protected, inflamed pocket.
And peri-implant diseases, infections around dental implants.
Yes, these are becoming increasingly important. Peri-implant mucositis, which is like gingivitis around an implant, and peri-implantitis, which involves bone loss around the implant, like periodontitis, often involve microflora in the peri-implant sulcus or pocket that is very similar in composition to that found in chronic periodontitis. It's typically dominated by diverse gram-negative anaerobic bacteria such as Fusobacterium spp, P. gingivalis, Prevotella spp, and sometimes A. actinomycetum comitans.
Similar players causing trouble around implants too. Now, moving inside the tooth again, what about dentoalveolar and endodontic infections? Infections of the pulp and root canal system.
These are also typically polymicrobial and endogenous infections, meaning they originate from the patient's own oral flora that gains access to the normally sterile pulp space, usually through deep caries or sometimes cracks or trauma. They usually occur when microbes colonize the necrotic pulp tissue within the tooth root canal system, that's endodontic infections, or spread from there, or sometimes from deep periodontal pockets into the surrounding bone, causing dentoalveolar abscesses.
And the microbes involved. Still the anaerobes.
Yes, the sources clearly state that obligate anaerobes consistently make up the largest group of causative organisms recovered from these infections. The necrotic pulp and abscess environment is perfect for them.
Which specific anaerobes are commonly isolated from infected root canals and abscesses?
Common isolates listed in the sources include Streptococcus species, Porphyromonas gingivalis, and P. endodontalis, Prevotella intermedia and P. melanogenica, another black pigmented species, and Fusobacterium nucleatum. Endodontic infections, referring specifically to infected root canals, are essentially described as complex polymicrobial biofilms adhering tenaciously to the intricate root canal walls. Primary endodontic infections are overwhelmingly dominated by obligate anaerobes.
Any non-bacteria commonly found in root canals?
Interestingly, yes. Candida albicans, the fungus, is also noted as the most common fungus recovered from endodontic infections, though often found more in secondary infections or persistent cases that haven't responded to initial treatment.
And the biofilm aspect is particularly challenging in root canals, isn't it?
Yeah.
Because it's hard to clean them out perfectly.
Absolutely. A crucial clinical point highlighted is that these polymicrobial biofilms within the complex anatomy of infected root canals have a higher endotoxin content, which drives inflammation in the surrounding bone, and they are significantly more resistant to antimicrobial irrigants and medications compared to planktonic bacteria. This is a major reason why thorough mechanical cleaning and shaping of the canals is so critical in endodontic treatment.
Makes sense. Finally, what about osteomyelitis of the jaws? Bone infection.
While less common than the other oral infections we've discussed, osteomyelitis, an infection of the bone itself, particularly in the jaws, is often a serious complication that spreads from an untreated dentoalveolar infection or sometimes a deep periodontal pocket or even following surgery or trauma.
And the microbes involved here?
Anaerobes are again the most common isolates in these cases, including species like Tannerella forsythia, Prevotella species, Porphyromonas species, and Fusobacteria. The sources also mention that osteomyelitis of the jaws can occur as a complication following radiation therapy to the head and neck area, which can compromise blood supply and host defenses in the bone.
Okay, so for you as a dental science student listening, this section really gives you the specific microbial signatures or the usual suspects associated with the key infectious diseases you will encounter and need to treat. Knowing, for instance, that an endodontic infection is likely a mixed anaerobic biofilm, or that the painful destruction of ANG is linked to that specific fusosporocidal complex, or that Aggregatibacter actinomycetum comitans is a prime suspect you should consider, in a case of localized aggressive periodontitis, directly guides your clinical thinking. It helps you decide which diagnostic tests might be useful and what treatment approaches, including potential antibiotic choices, would be most appropriate.
It really connects the microscopic world directly to the macroscopic clinical reality you will face every day.
Okay, to wrap up our deep dive today, let's look at the practical clinical considerations that the sources highlight when you're actually diagnosing and treating these oral anaerobic infections, particularly those involving those stubborn biofilms. What are the challenges?
Absolutely. The sources touch on several important practical points that you'll need to consider in practice. First, specimen collection is emphasized as crucial for accurate microbiological diagnosis, especially if you intend to send a sample for culture and sensitivity testing. This helps identify the specific organisms involved and determine which antibiotics would be most effective against them.
What are the best practices for collecting a sample, particularly when you suspect anaerobes are involved? They're sensitive to oxygen, right?
Yeah. They are, so meticulous collection is paramount. And it's essential to collect the specimen before initiating any antibiotic therapy, if possible, as antibiotics will obviously alter the microbial population present and make culture results less reliable. For an abscess, the sources strongly recommend obtaining aspirated pus using a sterile needle and syringe whenever possible.
Why aspiration rather than a swab?
Because aspiration from the core of the abscess provides a much more representative sample of the actual infection's microbes, avoiding surface contaminants, and it inherently protects the anaerobes from oxygen better than a surface swab. If aspiration isn't possible, a biopsy or curettage sample might be taken. Using specialized anaerobic transport media is highly recommended to protect the sample from oxygen exposure during transit to the laboratory.
Okay.
Specimens must be handled carefully to minimize oxygen exposure. The sources also specifically advise avoiding cotton swabs for anaerobic cultures, as the cotton fibers themselves can contain residual oxygen or substances that might inhibit the growth of some fastidious anaerobes.
Okay, so getting a good sample that keeps the anaerobes viable for the lab is more involved than just a simple swab. And once it gets to the lab, identifying them can be challenging too, right? You mentioned they're fastidious.
It certainly can be, which is the second challenge highlighted: identification. As we've mentioned, many oral anaerobes are fastidious, they have complex nutritional requirements, they're picky eaters, and are often difficult to culture using routine laboratory techniques.
What do they need?
They often require enriched growth media containing things like blood, hymin, and vitamin K. They need specific atmospheric conditions, usually an anaerobic chamber or jar with a specific gas mix, often 5-10% CO2, some hydrogen and the rest nitrogen, to create a very low oxygen or oxygen-free environment with a low redox potential. And they typically require longer incubation periods compared to many aerobic bacteria, sometimes several days or even weeks for some slow growers.
And this difficulty is why molecular techniques were such a game changer for understanding the oral microbiome.
Precisely. This historical difficulty in culturing a significant portion of the oral microbiota is exactly why molecular methods like sequencing the 16S rRNA gene or using specific DNA probes like in PCR assays have been so vital. They allow microbiologists to identify the presence and relative abundance of bacterial species based on their genetic material, even if those species cannot be easily grown on a culture plate in the lab.
So diagnosis can involve challenges with both collecting the sample and processing it in the lab. What about the challenge of antibiotic resistance? We know biofilms are resistant.
Yes, this is a very significant clinical hurdle, particularly as we've stressed throughout, because many of these infections are biofilm related. Organisms living within biofilms are generally substantially more resistant to antimicrobial agents compared to their free-living planktonic forms.
And testing for resistance takes time.
Right. While antibiotic sensitivity testing, determining which antibiotics will kill the cultured bacteria, can be performed on pure cultures isolated from a clinical sample, the results typically take 2 to 4 days, sometimes longer for slow-growing anaerobes, to become available. This lag time sometimes necessitates starting treatment with a broader spectrum antibiotic empirically based on the most likely pathogens for that type of infection before results from a targeted sensitivity test can guide therapy, assuming a sample was even sent and successfully cultured.
Which brings us finally to treatment. Given these diagnostic and resistance challenges, what does effective management of oral anaerobic infections typically involve? According to the sources, is it just about picking the right antibiotic?
No, the sources make it very clear that effective management nearly always involves a combination approach, especially for biofilm infections. Mechanical debridement is consistently highlighted as a key, often the most important step.
Mechanical debridement meaning physically removing the source of infection.
Exactly. Especially for infections where the biofilm is the primary culprit, like removing plaque in calculus in caries and periodontal disease, or thoroughly cleaning out the necrotic tissue and biofilm within infected root canals during endodontic treatment, or draining an abscess surgically. You have to physically remove the bulk of the infected tissue and the microbial mass.
So your skill with instruments and surgical procedures is just as important, if not sometimes more important, than prescribing medication.
For many of these conditions, yes, the sources strongly imply that mechanical removal of the biofilm and the necrotic source is paramount for success. It is used in addition to appropriate antimicrobial chemotherapy, which should be guided by knowledge of the typical flora involved in that specific type of infection, and ideally by sensitivity testing if available and warranted by the severity of the infection.
Any specific antibiotic sensitivities mentioned for these anaerobes?
The sources do note some general patterns, for instance, that oral spiricotates are generally sensitive to penicillin and metronidazole. Metronidazole is often effective against many obligate anaerobes, but not facultative ones. Penicillins, sometimes combined with beta-lactamase inhibitors like clavulanate, for example, augmentin or clindamycin, are also frequently used for mixed oral anaerobic infections, but resistance patterns can vary and are increasing.
Okay. So for you as a dental science student listening, these clinical considerations are incredibly practical. How do you collect a diagnostic sample properly, especially for those finicky anaerobes? Why are some bugs so hard to pin down in the lab? Why isn't simply prescribing an antibiotic always enough, particularly when you're facing those resilient biofilm fortresses?
Right.
And understanding why your hands-on skills in removing infected tissue, scaling away plaque and calculus, and thoroughly cleaning root canals are often the most critical components of treatment for many of these conditions. These are the real-world implications of the microbiology we've explored today.
The biological nature of these infections, especially their tendency to form biofilms in anaerobic environments, directly dictates the necessary clinical strategy. You have to address both the bugs and their environment.
So what does this all mean as we wrap up? We've taken a pretty deep dive into the complex, dynamic, microscopic world of the oral microbiome, guided by your sources. We've traced the path from a state of harmonious equilibrium, or symbiosis, to a state of dysbiosis, which we learned underpins the development of major dental diseases like caries, periodontal disease, and serious dental-alveolar abscesses.
We've specifically explored the diverse and clinically significant world of non-sporing anaerobes, from the notorious pathogens like Porphyromonas gingivalis and Aggregatibacter actinomycetum comitans that drive severe periodontal destruction and abscesses, to species like Fusobacterium nucleatum that are critical bridging organisms in biofilms and key players in necrotizing infections, and even potentially benevolent species like Veillonella that help modify the local environment.
And we've seen how these microbes predominantly live within complex, highly resistant biofilms, like dental plaque, and the significant clinical challenges this presents for accurate diagnosis and effective treatment. This really emphasizes the necessity of both thorough mechanical debridement to physically disrupt that protective structure and appropriate targeted antimicrobial chemotherapy where needed.
For you, as you continue your studies in dental sciences, this knowledge isn't just academic detail. It's absolutely fundamental. It forms the biological basis that underpins how you will diagnose oral diseases, assess risk factors for your patients, plan preventative strategies, and execute effective treatment plans in your future practice. You are learning to manage a complex, dynamic, living ecosystem within the oral cavity, not just treating individual teeth or gums in isolation.
And here's an important question, that this deep dive into the resilience and complexity of the oral microbiome, particularly within the protective fortress of the biofilm, really raises something for you to ponder as you think about the future of your field. Given how challenging it is to truly eradicate these established, resistant microbial communities with our current approaches of scraping and antibiotics, how might future innovations in dental science, perhaps through new technologies, maybe targeted molecular therapies or perhaps sophisticated ecological modulation strategies, move beyond our current methods? Could we find ways to better diagnose subclinical dysbiosis much earlier, before disease is obvious, or prevent that harmful shift more effectively? Or maybe even target these resilient microbial fortresses in entirely novel ways that we can't currently imagine? What's the next frontier in managing this microbial metropolis living inside our mouths?
That's a fascinating challenge, and definitely where a lot of research is heading, trying to be more precise and more ecological in our interventions. A great question for the future of dentistry.
Indeed. Well, thanks for joining us for this deep dive.
My pleasure. Until next time.